Well, I'd like to welcome everybody to our colloquium again. We took a week off for GSA, and you should talk to some of us about it. It was really a nice meeting, good to listen to talks. And while we had already invited Brittany to speak, it was nice to get to catch up with her at GSA last week. This afternoon at 4 o'clock, we're going to have Oktoberfest just outside. German beer, well, not German beer, but beer labeled Oktoberfest. Nothing pumpkin-y, but Octobery. And there will be bratwurst and other foods from 4 to 7 outside in the courtyard, the typical party place.
A little better, but I'm staying home and not going to work.
Hey, Greg. Hey, Greg. 2 weeks from today, pretty much everybody in this room is invited to go to our retirement lunch. One of our honorees is Claude Hersberg. Joining him will be Dennis Kent and families, George McGee and Martha Withjack. We will meet starting around 11 at the Rutgers Club on Livingston campus. It is fully all food provided, and it's a nice buffet. I'm choosing the menu as we speak, but show up around 11 and start eating 11:30, 11:45, and we'll have a little bit of a program honoring their contributions to us and a little bit to the scientific field. And it's just a way that we have of honoring those who contributed for a few decades to the betterment of our department. Are there any other announcements I should make?
Can you just tell everyone there are 2 people online that are still unmuted? Can you tell them to mute themselves?
There are 2 people online. Please check whether you're muted or not unmuted, but 2 of you are not muted right now, and Matt can't.
Rob Sherrill and Mark Feingenson.
Mark Feigenson and Rob Sherrill need to mute. Did that do? Well,
you'll know.
All right, let's get going. This is Brittany's time.
We're good.
All right, we're good. It's my pleasure to introduce Dr. Brittany Hupp today. Brittany, I followed from afar, first in the PETM. I've read her papers, and she worked on the PETM, which is of interest to several of us here. And then after getting a PhD at Wisconsin, working with Clay Kelly, she went to Oregon State on a NOAA climate change postdoc and was there 2 or 3 years.
1 year.
Oh, just 1 year.
Yeah, it's been a quick turnaround. Lots of cross-country moves.
And now she's moved to the East Coast. She's down at George Mason University as an assistant professor, and we'll be setting up a stable isotope lab where she will continue to make those measurements. She's from Ohio originally, has traversed the country in one direction, is back on it's on the East Coast right now. And with that, I'm going to turn it over to Brittany. Welcome.
All right, thank you for that introduction. I'm going to do a little Zoom magic here really quick. Maybe I...
Is that too much, Matt?
I think that's fine.
Okay, there we go. All right. There we go. All right. Thank you again for that introduction. So today I've come up with a very general talk, which gives me, or a very general title, which gives me the free range to talk about all kinds of fun Foram things that I've been working on over the past few years. So, novel geochemical approaches to interpreting past climate, oceanographic, and ecological signals in planktic or planktonic Foram records. We'll see here. Now, I recognize many of the people in this room are intimately familiar with planktic forams, but in case you are here to kind of broaden your horizons a little bit, we'll go over some planktic forams 101. So what are planktic forams? again, these are microscopic zooplankton. You can see two of them here on the nose of a penny. They are kind of first order consumers within the global ocean, so semi-foundational to these marine ecosystems. They're alive today but have an abundant fossil record that dates back to the Jurassic. And today I'm going to be talking about planktic forams. So again, these are critters that live within the surface ocean, either within the mixed layer where we have semi-homogeneous temperatures, or within the thermocline where we have those temperatures transitioning from surface to deep ocean temps. And as again, many of you probably already know, what's really great about working with these planktic forams is as they live within the surface ocean, they build these calcium carbonate shells that inherently end up incorporating geochemical markers that can tell us about the environment that they've been living in. So they're essentially excellent geochemical archives of ocean climate change, but equally as important is to recognize that they are also critters. So they are also responders to ocean climate change. So there's your quick Planktic Forams 101, but really for today, what I want to do is show you three kind of quick case studies that can show you how we can use one of these novel geochemical approaches to learn more about a given subject. And I'll be spanning anywhere from the PETM all the way to the modern. So first I want to give you an example of how we can use carbon isotope signatures of whole individual Planktic foram shells to learn more about how they responded to past periods of abrupt climate change. And in this case, not surprisingly, I'm going to be giving you an example from the Paleocene-Eocene thermal maximum, or the PETM. Again, something that probably several people are familiar with, but the PETM was a rapid global change event that occurred approximately 56 million years ago. We recognize the PETM in the global rock record by this distinct negative carbon isotope excursion, which is signaling the rapid influx of isotopically depleted carbon into Earth's ocean atmosphere system. So not surprisingly, with this carbon, we see ample evidence of rapid global warming on the order of 4 to 5 degrees Celsius on average across the global ocean. And again, with that carbon, we see ample evidence of ocean acidification, changes in hydrologic cycling, ocean deoxygenation, and biotic turnover, both especially within the terrestrial and within the benthic realms. So working with kind of my previous advisor at Wisconsin, well, again, the PETM, why do so many of us actually know about this? Well, even if you don't study the PETM, it's been argued because of all these various reasons here to be a decent analog for thinking about kind of the future consequences of modern climate change. So we were particularly interested in better understanding how did pelagic communities respond to the PETM. So how do we actually evaluate this? And when I say pelagic, not surprisingly, I'm going to be focusing on planktic forams, right? Again, several of us work on these cores, but just to talk about it more, how do we actually study these PETM pelagic communities? Well, one really great archive that we have is the investigation of deep sea sediments, most of which are recovered through programs like the International Ocean Discovery Program and its precursor scientific ocean drilling programs. And here I'm actually showing you a series of cores from the Walvis Ridge in the South Atlantic. And just looking at this, we can see distinct sedimentary changes from more carbonate to clay-rich sediments, marking the onset of the PETM and associated acidification. But if we were to focus more on these kind of carbonate-rich sediments and look at them under the microscope, we'd see thousands upon thousands, millions of planktic foram shells. So how do we actually use these records to learn more about kind of planktic foram community ecology? Well, working with a core like this, we can extract a stratigraphically restricted sediment sample, maybe 1 to 2 centimeters, split the sample until we have a relatively small random sample of shells, count and identify at least 300 individuals, and that allows us to kind of repeat this process for a series of stratigraphic samples and look at things like relative abundance of various species and changes in diversity. But let's take a moment to actually think about, we doing okay? Or is this?
Would you click on the X? Oh, you don't see it.
Oh, that X, that one, I see it up there.
Yeah.
Now I have to play the game where I have to try to see this tiny X. Oh, it's not recognizing it. Right?
Just go on.
Right? Oh, okay.
You're clicking in PowerPoint, not clicking in Zoom right now.
Oh, I see.
We'll ignore it.
Okay, don't worry, titles are important. We'll figure it out. All right, so with this in mind, this is how we actually interpret these community changes, but let's think about these deep sea records that we're working with. In thinking about these pelagic sedimentary records, we know that they have very low sedimentation rates that ultimately lead to pretty condensed paleoclimate records, particularly very rapid events like the PTM. These condensed paleoclimate records are highly susceptible to sediment mixing, whereby these microfossils can be easily displaced from their original depositional horizon by processes like bioturbation and winnowing by bottom water currents. And so we at least somewhat instinctively recognize that when we're looking at these microfossil records, we're typically looking at some sort of blended, time-averaged, aggregate assemblage. And it's possible that when we're looking at these very rapid global events, that this time averaging could be masking the biotic response of these communities to abrupt climate change, like the PTM. So this leads to the second leading question: how can we deconvolve the effects of sediment mixing on these microfossil assemblages to kind of better recognize how they're responding to these abrupt climate change events? And with that, this is where I want to tie in those geochemical approaches and introduce an approach that we're calling isotopic filtering. And to do this, I'm going to be showing you a case study from Ocean Drilling Program Site 865 in the equatorial Pacific Ocean. But when we think about the PTM, there really are only so many deep sea PETM sites. So it should not be surprising that the planktic forum assemblages from this site have already been evaluated and were actually published on in the late 90s in a series of papers. We're going to go with the black and then the bright white to keep you all awake as we transition through this talk. So the planktic forum assemblages from this site were already evaluated in the late 90s. And in fact, again, because we only have so many deep sea records of the PTM, studies like this can really help to kind of shape our thoughts on how these communities responded. So there were a few key observations that kind of came out of these series of papers. First was a rapid diversification of planktic foraminiferal communities. And this is in part due to kind of the influx of what has become known as excursion taxa during the PETM. So also within one of these papers, this new species, Morsavella allisinensis, is one of these excursion taxa that essentially evolved during the PETM, only existed for that short amount of time, and then disappeared from that record. Now, one kind of lesser known aspect of these papers is that in addition to kind of looking at community ecology and describing that new excursion taxa, they also did about 200 single shell isotope analyses from the CIE, or carbon isotope excursion interval, just on a few different species. And here we're going to actually look at the distribution of that data. Now, thinking about this, if we're taking all of our foram shells from this CIE interval, you would hope that they are all recording values somewhere between 1 and 2 per mil. But when we look at this distribution, we see that it's clearly bimodal, where one mode is clearly recording the CIE values, telling us that these individuals were actually present during the PTM. But we also find this second mode, which records distinctly pre-CIE values. So again, semi-intuitively, it's not surprising that we see that these assemblages that exist within the carbon isotope interval marking the PTM are blends of pre-CIE and CIE forams. And so this leads to a few questions. How mixed actually are these assemblages? Has sediment mixing potentially biased our perception of the biotic response of these communities to the PTM? And is it possible that we can leverage these carbon isotope signatures of individual planktic foram shells to potentially deconvolve these taphonomic versus biotic signals within this record? And that's essentially what we set out to do. So in order to actually do this isotopic filtering approach, it was important to kind of recount the assemblages Although the assemblages had already been published, I counted from a much larger size fraction, which was the same size fraction that the isotope analyses could be performed at. Some of the previous work I've done has kind of shown how different size shells can be more easily reworked, so it was really important to keep things pretty consistent size fraction wise. So in doing this analysis, we recognize about 24 different species, but these 9 shown here make up the vast majority of the pre-CIE and CIE assemblages, about 82 to 94%. So these were the taxa that we were going to kind of target for abundant single shell isotope analyses. And so what we essentially did is across 5 stratigraphic samples within the CIE interval, we did hundreds of isotope analyses across these 9 different taxa. And here I'm showing you the distribution of all of those single shell isotope analyses. So between the previously published and newly conducted analyses, we did about 548 in total that we were working with, again, across these 9 taxa, across 5 different stratigraphic samples. And just looking at the data here, what we see is that only about 50% of all of those shells actually record CIE values. And in fact, about 49.5% are pre-CIE contaminants that have been reworked and now redeposited within that CIE interval. But we can take this data and now start to look at it separated by those 9 major taxa. And when we do that, there's a few patterns that we can pull out. And just to again orient you, we're looking at these histograms of carbon isotope data for the CIE and pre-CIE. But again, all of these shells were collected from within that CIE horizon. So when we look at the data in this way, we can kind of separate these 9 taxa into 3 different groups. First, we have the CIE taxa, where you can see their single shell isotope analyses all kind of fall within those CIE values. Conversely, we have some taxa that exist within the CIE only as reworked contaminants, as shown by their pre-CIE carbon isotope signatures. And then we have some taxa which are a mix of in situ and reworked specimens existing within the CIE. So now we can take this information and look at it within its stratigraphic context. So again, this data is from 5 different stratigraphic samples from within the CIE interval. And I'm plotting the proportion of reworked specimens per taxon. So for example, here in A, looking at Subatina, the thermocline dwellers within this group, we see that 100% of their shells recorded pre-CIE values, telling us that they were 100% reworked within the CIE interval. And in looking at the data in this way, we can again kind of divide these taxa into 3 separate groups. So first we have the Lazarus taxa, and these are critters that were present before and after the event, but either disappear for the entirety or part of that CIE interval. We also have the holdover taxa. These are the few critters that made it all the way throughout the event, so present before, during, and after. And then we also have the excursion taxa, which were really only present during the PETM and show 0% reworking, no surprise. And so now the idea was to take these kind of estimates of reworking, remove those proportions of rework contaminants from the assemblage counts and reevaluate the assemblage, comparing kind of the unfiltered traditional point counted assemblage to now this isotopically filtered assemblage. And here we are looking at that data. So looking at the unfiltered assemblages, we can see why, here we go. We can see why the previous interpretation was this distinct increase in local diversity. We see the influx of these 2 excursion taxa shown here in gray and white, and a relative change in the relative proportions across the other major taxa. However, when we actually go and remove those reworked contaminants from this assemblage, we get a very different story, where essentially these communities collapse to being composed of only about 4 taxa within the early CIE interval. So a very, very different story. There's a few kind of key results that we can take away just by looking at these assemblage comparisons. First, it's clear that planktic foraminiferal diversity did not increase in the tropics during the PETM, but in fact clearly decreased. And when we think about the fact that we're looking at this tropical site, it should not be super surprising that we see this. There's been other studies that have recognized heat stress signs in other plankton types within the tropics, so it's not surprising that these planktic forams also maybe didn't like those extreme temperatures. Additionally, we can look at things not just on the community level, but we can start to look at how individual taxa responded to the event. So here I want to show you an example of these two taxa, Morsavella aqua and Sabotinae, as well as the Sabatinids, shown here in pink. And if you look in the pre-CIE interval, they account for nearly 50% of the entire assemblage. However, they both disappear from this site during the majority of the CIE, only to reappear during the recovery. And so we can basically take this as a reflection of extratropical migration by these species that were really quite prevalent in those pre-CIE assemblages. And so using this isotopic filtering approach at Site 865 has shown us that tropical planktic foram communities were actually highly sensitive to PETM warming and acidification, contrary to kind of previous interpretations. And this is indicated by evidence for extratropical migration, as well as a distinct decrease in tropical diversity. But what we can also see by using this approach is that it essentially yields unprecedented resolution into the biotic response of the PETM at this deep sea site. And hopefully this approach can be used to evaluate other PETM sites, as well as other ocean climate events that are marked by distinct biogeochemical excursions. And so hopefully that gives you an idea of how we can again use these whole shell, single shell values of planktic forams to learn more about how they responded to these past periods of abrupt climate change. But now I want to show you how you can, instead of using whole shells, use, oh, see, you missed the important part right there, in situ delta 13C measurements. So not whole shell, now we're going to go even deeper, even tinier, in situ delta 13C measurements of PETM foram shells to learn more about carbon cycling during past periods of abrupt climate change. And so I've been talking about using these delta 13C records, but now I want to think more about carbon isotope records and stratigraphies and what they can kind of tell us about events like the PETM. Well, these types of records, they can provide important constraints on things like climate sensitivity in Earth's past, especially when they're coupled with other proxy records that allow us to evaluate how much warming occurred in response to what type of increase in PCO2. We can also start to make interpretations about the rate of carbon injection into the ocean atmosphere system. And we can also start to make interpretations, or at least hypotheses, about the likelihood of different carbon sources. And so one way in which we start to maybe approach some of these questions is by thinking about the magnitude of carbon isotopic change during an event like the PETM. And although I've been kind of inherently talking about this, just as a quick review, when we look at a carbon isotope stratigraphy, we're looking at kind of the pre-carbon isotope excursion, then we have our CIE, or carbon isotope excursion, kind of main event section, and then we typically have some sort of recovery, at least when we're looking at these deeper sea records. And so, as I was saying, one kind of feature of these stratigraphies that we can be particularly interested in is the carbon isotope excursion magnitude. So what is the magnitude of isotopic change that we see at the onset of this event? And one way in which we kind of calculate this is by comparing the pre-CIE average to whatever kind of our CIE minimum is for a given stratigraphic sample. So in this case, we're looking at a bulk carbonate record from Ocean Drilling Program Site 1135, which is in the southern Indian Ocean on the Kerguelen Plateau. And so for this bulk carbonate record, we could say that the CIE magnitude is about -2.4 per mil. Now, how do we actually use a value like that to start to make interpretations or model different carbon cycling scenarios? Just to give you an idea of how we can use this magnitude is we can take this CIE size, essentially shown here, and we can start to think about potential sources of carbon that would have been tied to this event. So for example, we could say, what if it was dominantly methane, which on average probably has a signature around -60 per mil, that caused the PETM? I'm not committing to that, just as an example of a hypothesis that could be possible, right? Well, we can start to look at our carbon isotope excursion size and our proposed signature of a potential source, and look at this line and start to estimate the amount of carbon, or specifically the amount of methane that would be necessary to cause that amount of isotopic change. Easy peasy, right? Why do we need to be worried about what the size of this excursion is? This bulk carbonate record told me it's -2.4 per mil. Well, it's not that simple. And in fact, when we actually look at several different PETM records, we get pretty different signatures in terms of the size of these carbon isotope excursions. Just very broadly speaking, if we compare average marine records to average terrestrial records, we find that in the marine realm, our CIEs are averaging about -2.8 per mil. However, in the terrestrial realm, we get much larger magnitudes of about -4.7 per mil. So you can see how these discrepancies in the size or magnitude of the CIE can lead to pretty big differences when we're thinking about the amount of carbon that could have caused this type of event. And so this kind of leads to this next leading question: why are the CIE magnitudes so much smaller within these marine records when compared to terrestrial PETM records? And if we really look at what makes up the vast majority of these marine records, we're really talking about various types of carbonate records. So let's think about what these carbonate marine records can be subject to. Well, we've already talked about sediment mixing, right? We've seen how those can be affected by sediment mixing. Dissolution, which is definitely something to consider during an ocean acidification event, and diagenesis. So is it possible that these secondary mechanisms are what's causing these kind of attenuated carbon isotope excursion magnitudes that we find in marine PETM records? Let's kind of go through each of these. So we've already talked quite a bit about sediment mixing, but let's talk about it more within the context of this specific PETM site. So here I'm showing you the same bulk carbonate record that I showed you just a little bit ago. And we've already decided that this excursion magnitude is about -2.4 per mil. But this is a bulk carbonate record, and bulk carbonate records are composed of all kinds of different carbonate, of all kinds of different sizes, and they can be highly susceptible to sediment mixing. So maybe we can take the same approach that I just showed in kind of my previous mini talk and look at single shell Foram isotope analyses to see if we get a different excursion by essentially, again, deconvolving some of those effects of sediment mixing. So here I'm showing you a single shell Planktic Foram record from this same site. And when we look at the excursion magnitude within this single shell Foram record, we get a much larger magnitude of about -3.3 per mil. So we've been able to at least remove some of those kind of reworked contaminants and really look at the isotopic change between the pre-CIE and CIE minimum. What about dissolution? Well, bad news about dissolution: if part of the record's dissolved, unless we're building a time machine, we're not getting it back, right? We know that at the onset of the PTM, we see lots of evidence for ocean acidification, and so it's not surprising that many of our deep sea records experience some sort of truncation due to this dissolution. However, when one thing I will say at least is when we look at terrestrial records, we do see the same kind of rapidity of isotopic change that we see in the deep marine. But again, what are we going to do if it's dissolved? It's gone. Well, what about diagenesis? Typically when we think about carbon isotope records, we don't really think diagenesis is a huge problem or something we need to think about. But is it possible that diagenesis could be further contributing to these attenuated excursions? And if that were the case, how could we potentially detect it? Well, let's take a moment to think about how something like this single shell Planktic Foram record is constructed. Typically when we do these Planktic Foram records and we're doing one or multiple shells, we use gas source mass spectrometry, where again we're taking one or more of these shells and dissolving them in phosphoric acid and ultimately measuring the carbon and oxygen isotopes from the CO2 gas that is released. So we're taking that whole shell, anything related to it, and measuring from the CO2 gas that is released from that reaction. But let's actually look at what some of these planktic forams look like from this interval. One kind of key feature that we find in a lot of planktic forams from the PETM is that many of them, since they're pretty much all porous, right, have these distinct features called muricae, which are essentially bumps that form on these interpore ridges. And although these muricae appear to be really part of the foram's morphology, they also act as really great centers for kind of nucleation and growth of secondary calcite. So here we're looking at some morsavelids, and we can see these distinct muricae blades that pretty much look like if you were going to inorganically precipitate just calcite crystals, right? So let's again look specifically at some of these shells from this 1135 record. And not surprisingly, because this is like 90% of the species that were alive at this time, they do indeed have muricae. But another thing about this site that's interesting is it's also a very chalky site. There's a lot of calcareous nanofossils, and so when we look at these shells, we can see that they're kind of encrusted in these calcareous nanofossils. And in fact, when we look inside of them, it's not surprising that a lot of these chambers can be filled with calcareous nanofossils that may or may not be easy to actually remove from those chambers. I think it's also important to think about when we're thinking about this kind of chalk or fine fraction carbonate that exists, there's really no guarantee that that fine fraction carbonate is actually from the same time period in which the foram shell precipitated. So again, thinking about kind of these gas source records, we're taking this entire shell and whatever's in it, whatever's on it, depending on how good you can clean it, right, and dissolving it. So is it possible that we could leverage some sort of in situ methodology to kind of investigate this potential for diagenetic overprinting? So again, I've already shown you how with this gas source mass spectrometry, we're taking this whole shell, everything in it, on it, and dissolving it and measuring the isotopes from that CO2 gas. But there's another opportunity that we have to measure these carbon isotopes, and that's using SIMS, or secondary ion mass spectrometry. And this is where we're actually measuring the carbon isotopes from secondary ions that are sputtered from the surface of a given sample of a relatively minute domain. So for example, here we're looking at one of these shells in cross section, and this little black dot here and here, those are two SIMS pits. And so these pits are about 10 microns in size to give you an idea of what we're working with. And so the idea here was to, well, we've already established a bulk. Well, the bulk record was partially established, but we have a bulk record. We established the single shell gas source planktic foram record. And the idea was to go forth and establish a SIMS record for this 1135 site and see if we end up finding a larger magnitude. Also, because of these very small kind of pit sizes that we have to work with, we can also evaluate intrashell delta 13C variability and see if we see differences between our inner chamber walls and those outer muricae blades. And so I'm actually going to show you some of that intrashell data first. Here I'm showing you an example of one of these acarininids. Yes, okay. And here we're looking at the cross section here. You can see there have been 4 different SIMS pits that have put into this shell. If you are familiar with SIMS, or if you're not, it's very tricky. If you are not okay with failure, don't do SIMS. But here we have at least a few pits that kind of worked out. And so we can compare the signatures of those inner chamber walls to those outer muricae and look at these relative gradients. And so here what I've plotted is whether we find a positive gradient, inner minus outer, like what we see here, or no gradient, there were no negative gradients, between shells within the pre-CIE, CIE, and recovery intervals. And essentially what we find, even though our N is not huge, is that within these shells, we do typically find differences in their carbon isotope values between the between the inner chamber wall and their muricae. And we end up finding these more positive inner-outer gradients specifically within the CIE interval. Why is that the case? Why do we see these differences, and why do we only really see them during the CIE? Well, let's consider what's happening in terms of ocean carbonate chemistry during the PETM. Prior to the event, we can say we have a relatively normal, lysocline, where again these muricae exist on these shells just as general features. So we have shells that are living in the surface ocean, they die, they sink to the seafloor, they become part of our deep sea sedimentary record. During the PETM, at the onset, we get carbonate undersaturation, which leads to the dissolution of some of that pre-CIE material, as well as deposition and dissolution of PETM or CIE material, which kind of leads to this mixed dissolved inorganic carbon pool within those pore waters. And then we start transitioning into the early recovery, and we get this carbonate oversaturation, this overshoot, which allows for kind of more carbonate precipitation and extended muricae growth on those CIE forams from this kind of mixed DIC pool. That's one way, at least, you could explain how this could happen. And so, like I said, with SIMS, we're trying to see if we can get a larger excursion magnitude by kind of removing any influence by those muricae blades and only looking at analyses that come from those inner chamber walls. And when we do that, we do in fact find a much larger excursion magnitude of about -5.3 per mil, which is inherently a lot larger than what we find in our bulk or gas source records. And notice here that really the forams are being affected again within that CIE interval because of the inclusion of these muricae blades. And so in terms of kind of bringing this back to this whole marine versus terrestrial differences, you'll see that this is much more kind of on the order of what we find within the terrestrial realm. So hopefully here you can see how kind of comparison of these whole shell versus in situ carbon isotope records show that indeed we do find much smaller magnitudes when we are looking at bulk carbonate and whole shell gas source delta 13C records. And this is due for a lot of different reasons. We see mixing of pre-CIE and CIE materials when we're looking at the bulk carbonate inherently. We see how CIE forams could be infilled with non-CIE fine fraction carbonate and included in those measurements. And we know that these muricae blades can really be diagenetic calcite that could also be included in these gas source records. We see that we have a much larger excursion within our SIMS-based record from Site 1135, which is more consistent with what we find in the terrestrial realm. And perhaps most importantly, taking a step back, we really don't think about diagenesis when it comes to carbon isotope records. We really attribute that to differences that we see in oxygen isotopes. But hopefully this is at least one example that shows that we should consider the potential effects of diagenesis on carbon isotope records, particularly when they are associated with periods where we have rapid changes in oceanic carbonate chemistry. So here again, hopefully you can see how now using these in situ delta 13C measurements, which is kind of pretty new in the world of forams in general, we can potentially learn more about carbon cycling during the PETM. And so now I want to move away from isotopes and think more about how we could potentially use in situ trace elements within modern planktic forams to better understand vital effects and how we should consider them as we work to build these paleo reconstructions. So I've talked a lot about isotopes, but here just as a reminder, trace elements are also incorporated into the calcite lattice of a foram shell as it grows within the surface ocean. And these trace elements within the shell are controlled by things like the trace element composition of the seawater, as well as the physical and biological conditions under which these shells are growing. And so because of this, there's been a lot of effort over the past few decades to really quantitatively constrain relationships between the trace element chemistry of these planktic foram shells and how that relates to their living environment, so that we can use those relationships to really probe paleo records. Probably one trace element proxy that we think we know super well or we feel the best about is magnesium calcium as a paleo temperature proxy. As many of you have probably already learned, as these shells grow under higher temperatures, they will inherently incorporate more magnesium into their calcite lattice. How do we actually establish a relationship like this? Well, there's several different ways, but just to give you an idea of a few, One way is through core top studies, where we go and collect these foram shells off the seafloor and compare their chemistry to kind of regional sea surface temperatures, assuming some sort of given settling rate. Not surprisingly, this definitely has issues. I've just talked about how the seafloor can be affected by sediment mixing in early diagenesis. And so this can be hard to create these relationships through this kind of methodology. Another way that we use quite a bit is through culturing studies, and this is where we go out and we collect these forams from their living environment, bring them back to the lab, analyze their chemistry, well, grow them under controlled conditions, and then look at their chemistry. However, it can be pretty difficult to replicate the natural living environment of a foram, and so sometimes growth in culture is not always reflective of growth in their natural living environment. And I want to give you one example of this. So if we're looking here, these are all modern species. And if we look at this top row, we're looking at these shells that essentially have not gone through this crusting process, which is where many of these species will grow these distinct crusts on their shells just after reproduction and just prior to death. So these are essentially fully mature forams. You don't see their cytoplasm because the shells have already been washed, but you can see that they look relatively thin. Conversely, these are recently deceased shells collected from the exact same plankton toes that have gone through this crusting process, and they look much more robust. And so when we grow these species in culture, it can be really difficult to get them to actually complete their life cycle and grow this thick crust. However, in nature, this is just part of their natural life cycle. And so when we look at paleo records, this is dominantly what we're finding. But when we're constructing these culturing calibrations, this is what we're dominantly using. And so this leads to a couple questions. Does the trace element content differ between these crusted and uncrusted shells? And if so, how does this actually influence reconstructions that are based off of culture calibrations of uncrusted shells? So to test this, we decided to collect pretty large populations of crusted and uncrusted shells of the same species that were all living within the surface ocean this past May. To give you an example here, we're looking, there's a lot of alive forams in this toe, but just looking here you can kind of see a nice adult foram with this nice pink cytoplasm next to a foram that has recently died and undergone this crusting process. And we brought them back to the lab and analyzed them using laser ablation ICP-MS. So laser ablation is a really great tool if you're interested in kind of intrashell variability with forams. It allows you to measure several different elements simultaneously. You can kind of alter spot sizes to work with different sized specimens. And what's really great is, in addition to being able to kind of calculate a whole shell value, you also get the elemental counts as you are actively ablating through the shell wall. And so all of these features kind of allow for further intra-shell investigation of trace element variability. Oh, I had this cool video, but we're going to keep on keeping on. And so we took these kind of populations back to the lab and used laser ablation to assess both kind of their whole shell geochemistry as well as intrashell variability of 6 elements: magnesium, sodium, strontium, barium, manganese, and zinc. And first I'm going to show you differences in their whole shell trace element geochemistry, specifically, again, between these alive, you can't see that, but you can see this. Between those alive uncrusted shells, which the data is going to be shown here in pink, and dead crusted shells, which is going to be shown in blue. And if we look at magnesium, there's not huge differences between the alive and dead shells. But if we look at our other elements, we actually find that several of these elements show statistically significant differences between those uncrusted and crusted shells. And so we're seeing that uncrusted shells tend to record higher trace element compositions than crusted shells, again, collected from the surface ocean from the exact same toe. So is it possible that these differences in trace element chemistry between these crusted and uncrusted shells is related to their intrashell variability? Or more specifically, is it possible that these crusts that are being added to these shells are pretty depleted in trace elements? Like I said, laser is great because in addition to kind of getting a whole shell value, you can see how the chemistry changes as you are ablating from the outside of the shell through the inner shell wall. So here, again, moving from the outside to the inside, we could generally say that the kind of first 50% of these spectra is representative of the outer calcite or the crust calcite, and that this inner 50% is kind of the inner shell wall. There we go. So this is just for one specimen, one crusted specimen. And if we look here, we'll see that many of these elements are more enriched within kind of that inner calcite relative to the outer calcite. But this is one shell. How does this actually scale up when we look at an entire population? And so in order to do this, essentially what I've done is not just eyeball 600 different spectra, but instead take the average of kind of the outer 50% of each spectra and compare it to the average of the inner 50%. So if they're somewhat the same, they'll plot along this one-to-one line. If we see that the inner calcite is indeed enriched, we'll see these populations sitting above the one-to-one line. And if the outer crust calcite is more enriched, we'll see them sitting below the one-to-one line. So here, if we look at all these plots, which you do not need to mentally absorb them whatsoever, we're looking at the data for each of the 4 final chambers for magnesium, sodium, and strontium. And I want to clue your eye in here to show you that for sodium, calcium, and strontium, calcium, many of these populations sit above that one-to-one line, especially as we look at progressively older chambers. When we look at our other trace elements with barium, manganese, and zinc, we see the same thing, where again those trace elements are higher within the inner calcite. So one last way we can kind of look at this data is, instead of just looking at it on these one-to-one plots, we can now actually subtract the outer 50% from the inner 50%. Again, if they're relatively similar, they'll sit along this 0 line. If there's positive values, that kind of further confirms that the inner calcite is recording higher trace elements. And so when we look at the data in this way, we can see that some trace elements like magnesium, manganese, and zinc are lower in crusted shells, specifically because their crusts are depleted in trace elements. And we know this because the alive shells like to sit along this 0 line, but in those crusted shells, we see this distinct inner-outer gradient, which leads them to have kind of a positive inner-outer delta difference. However, with our other elements, sodium, strontium, and barium, we see that the inner calcite will record higher trace elements regardless of if they've gone through this crusting process. And again, we see that because both our alive and dead shells that have either gone through this or haven't gone through this are sitting above that 0 line. However, it's worth noting that the differences are higher typically for these crusted shells in terms of intra-shell variability. And so these kind of comparisons in terms of trace elements for the crusted and uncrusted shells show that in general, dead crusted shells tend to exhibit lower trace elements than fully mature alive uncrusted shells. And we find that this is significant when we look at sodium, strontium, barium, and manganese. Differences between the crusted and uncrusted shells are often due to depleted or trace element depleted crusts, though there are some elements where we just find higher trace elements within the inner chamber wall regardless of crusting. And so I asked this question, which I asked at GSA. You know, you might see this and say, who cares? These are very fiddly observations related to these forams. But bringing it back to kind of how I started this last section, the key point here is if we're going to use culture calibrations that were established from uncrusted shells and we apply them to these crusted fossil records, it's going to inherently lead to kind of attenuated paleo reconstructions. And so I know I've talked a lot today, so I'll wrap it up, but I'm hoping that throughout this talk you can see why I went with a very general title, so I can tell you about lots of fun different ways that we can use different geochemical techniques to really dig deeper into how we interpret these Planktic foram records. Lots of great people to acknowledge in terms of collaborators, labs, crew, but I've been talking way too long, so I thank you and I welcome any questions.
So with the first part of your talk, so you saw the bimodal distribution of the C13. Do you see it in the O18 as well? Because, 4 to 5 degree warming, the surface layers, you might be able to detangle them similarly.
Yeah, so just to repeat the question was when we look at the bimodal distribution of carbon isotope signatures in PTM records, like what I showed here, do we also see it in oxygen? And the answer is sometimes yes, but in some of these cases, like at this specific site, site 865, there's not a large oxygen isotope excursion, and part of that is due to diagenesis. Yeah, but you could, like if you had like a very distinct, that's kind of a future thing I'd like to do, is try to apply the same approach, but focusing more on like oxygen isotopic change. Yeah.
Thanks, Brittany. That was really interesting. I was thinking about the second part of your talk when you saw an offset between when you looked at SIMS and you saw an offset between sort of the forum interiors and the Muricae.
Yes.
And you related that to changes in carbonate saturation.
Yeah.
I was wondering, would a test of that idea, if you could look at sites with different paleo water depths, would you expect to see like differences in the tempo of that, or differences in the magnitude of the offset?
Oh, that's a good question. Let's see, if you were looking at different, I think in theory, yes. I'll say this, so in terms, to add further context to this, with kind of the SIMS project that I was talking about, what's essentially been done is there haven't been a ton of people who have looked at kind of the Muricae difference, so how those kind of outer blades compared to the intercalcite. But there have been kind of interestingly unique sites where we find these distinct crystallites that kind of literally teeny tiny calcite crystals that precipitated, and they're only found within kind of the PETM interval. And so there's been kind of analyses of those, which also interestingly show kind of these intermediate signatures between the pre-CIE and CIE. But in terms of doing more kind of Muricae, I mean, I would assume yes, depending on where you are, you could find potentially larger differences, right? But I don't know if it would be directly tied to paleo water depth, I guess, when you're looking at the carbon isotopes. But I don't know.
I guess as even thinking about the tempo, I was thinking if you had in shallow water depth, maybe the lysocline reaching sooner, maybe in the first stage.
I see.
I don't know.
Yeah. Well, I feel like tying into your paper, right, how we've seen like larger excursions in shallower water depths, but that's more of a shelf perspective. So I don't know. It's a good thing to think about. Thank you.
Chris?
So this is almost more of a biology question, but it was something that you mentioned earlier and that I was then thinking about with regards to biological reactions to environmental changes. And I don't know why I'd never thought about it before, of the sort of idea of when the local environment becomes acidic, is it a local die-off of the population there, and then when the conditions are good, that population that remained rebounds, or do critters from elsewhere migrate back in? Like, what was this process, and also how does the behavior of these plankton affect, how active of a role do they play in leaving this area, coming back, and affecting the record as well?
Yeah. So, I decided to show this data, which is from a beautiful equatorial site, because if they disappear, you're like, oh, they must be leaving the tropics, right? Like that intuitively makes sense. But I've also done this isotopic filtering at a high latitude PTM site, which isn't published yet. But interestingly, we see the same thing where they're leaving. But where are they going? Is it due to local acidity conditions? I don't know. It's tricky. That's why you talk about the equatorial stuff, because then it's like a little bit more straightforward or a little bit more intuitive in terms of what would be affecting it. But another thing to think about is like we're talking about plankton, right? So there's not like intentional moving, but you know, how does dispersion play into this? And even like dispersion and current changes associated with the PTM, right? It's super complex and super deep time, which means I can do a lot of this if I really want to. But no, that's a good question and a good thing to think about. I don't know if I really answered your question, but
It was an open-ended,
yeah. It's pretty complex. Lots of things to think about. Yeah.
Matt?
Yep, we have one online. Vera's wondering, are the changes in TECA just from encrusting after death? There seems to be a periodic change in the TECA through the calcite test.
Yeah, no, they are not only controlled. So that's one thing when I was kind of doing those comparisons of inner outer, right? That generally gives us an idea of if the crust has higher or lower trace elements compared to the inner shell wall, but that totally eliminates the fact that there's all kinds of different intrashell variability that exists, right, in terms of banding due to periods or more like growth periods essentially. So no, it's not only related to crusting whatsoever. And I think there's a lot of effort. So a lot of that work that I'm showing here was done while I was at Oregon State. And there's a lot of people kind of working to better understand intrashell trace element variability and what is really kind of controlling those within modern forams. So this is just one example in terms of what to think about with trace elements, but depending on what you're trying to look at, there's a ton of different, I don't want to say triggers, but things that end up affecting intrashell trace element patterns within these shells. And I will say that one thing that we're kind of seeing is that it can be very different from species to species, especially spinos versus non-spinos. Very big differences.
I find both your PTM examples pretty compelling in terms of understanding what plankton actually responded. We've always kind of assumed we see the planktonic signals and the benthic signals are similar and different than the terrestrial. So you're now saying that planktonics are fitting the terrestrial pretty well, but since the 800-pound gorilla is the deep sea in terms of carbon storage, anyway, 36,000 gigatons versus a few thousand, would you speculate that the benthic signals are in fact correct, and would you want to go back and test that? So we see 2.75 plus or minus 0.5 in benthics.
Yeah.
So that is the carbon cycle in fact in terms of volume, if it's correct.
Yeah. Oof. I don't know, but then I feel like with that maybe, I don't know. I was going to say maybe dissolution plays more of a role, but it really doesn't. I don't know. That's a hard thing to pin.
You don't have all the,
Yeah.
I never liked working on because they're very complicated.
Yeah.
Okay.
They're pretty fiddly.
A lot of moving parts and
Yeah.
very complex lifestyles, and so it's actually interesting to see your response. But I was just little bottom dwellers are pretty simple.
Yeah.
Anyways.
I almost wonder though with like the turnover we see in the benthic realm, if there could be some weird dynamics in terms of like maybe even more pronounced extinction events that we aren't really like picking up on, but we're getting things that are early recoverers. And so I don't know, are we even still seeing a realistic amount of isotopic change in the benthic realm? I don't like just thinking about benthic turnover and also how these records can be affected by like things like dissolution.
And we know there's a lot of dissolution.
There's a lot of dissolution, right? But like I said, what are you going to do about it at the same time? So I don't know if I would hang my hat and say benthic is correct, but that's coming from a planktic person. So still things to think about. Oh, another thing, now I'm thinking about this, is also we see a lot of size changes in benthic forams during this time, right? And some of the other work I've done has kind of, like I alluded to, is that different size shells have more or less mixing potential, which I think also plays into it. There's like the whole extinction probability, but then there's also mixing and there's dissolution, and I'm not confident to say yes, the benthic is like truly recording the best record. But I don't know. Ask me in 10 years and maybe my answer will be different. It will hopefully probably be different. So.
Well, if there's no other questions, let's thank Brittany one more time. She will be around at Oktoberfest and around during the morning, tomorrow morning, if you would.